DNA, and so it includes a promoter, a regulatory gene, and then in the TRP operon, there is a promoter which has RNA polymerase and an operator, and then these are 5 genes that are of this operon. So in this section, the regulatory gene codes for an inactive repressor, and so in this situation, tryptophan is absent and the repressor is inactive, so the operon is on. So the RNA polymerase is able to transcribe these genes into these polypeptide subunits that are able to make these enzymes, which then code for tryptophan. In this situation, the regulatory gene codes for the protein, the same protein, but in this case there's an accumulation of tryptophan, so the tryptophan attaches to this protein, and so now it's active, it changes shape, and it then is able to connect to the operator. So in this situation, RNA polymerase is stopped, so our transcription cannot occur, so no RNA is made. Prokaryotic transcription. This is a DNA sequence from E. coli, and this DNA sequence includes a LAC operon, which has the regulatory gene, which is called LAC1. And so this regulatory gene codes for an active repressor. So when this repressor connects to the operator, transcription is not able to occur because the RNA polymerase is stunted and it's trapped, so the operon is off. In this situation, there's the presence of lactose, so allolactose, which is an inducer, is able to attach to the once active repressor, and it makes it inactive. So the inactive repressor is no longer able to stunt the transcription of this DNA sequence. And so now we can see that LAC-G, which is a gene that codes for beta-galactosidase, helps to hydrolyze lactose into glucose and galactosidase. And then permease is a membrane protein which is coded from LAC-Y, and this membrane protein transports lactose into the cell. And LAC-A is the gene that codes for transacetylase. Transacetylase has no clear function in the metabolism of lactose. A number are examples of prokaryotic transcription, and so in this whole example, lactose is present, so the repressor is inactive. That means that transcription is able to occur. But what we're considering now is the levels of glucose. And so when glucose is scarce, there are high levels of cyclic AMP. And so the cyclic AMP activates an activator protein called CAP. So once this activator protein is activated, it binds to the CAP binding site, which is located within the LAC operon. And this allows transcription to happen much faster, so you have a more abundant product of the enzymes that are used to metabolize lactose. But when glucose is present, there's lower levels of cyclic AMP, so the CAP will actually stay inactive, and it will be less likely that the RNA polymerase will bind to the DNA sequence. So it will be less likely, and there will be less product of the enzymes that are used to metabolize lactose. In eukaryotic transcription, when the DNA is first transcribed, pre-mRNA is made, and in order for the mRNA to be suitable for translation, it must first be processed in RNA splicing. So here's the pre-mRNA with its 5' cap, exons, and introns. And so the spliceosome consists of small mRNAs called SNRPs, and it's a complex of the proteins and the SNRPs. And so what it does is the small mRNAs act as, they recognize the splicing site on the intron as they base pair with specific sites on this intron. And so they also catalyze the splicing reaction. And so after the splicing reaction occurs, the intron is then released as the exons are joined together. So here's the released intron, and also what is also released is the spliceosome components, and then mRNA is made. Okay, here's a model of the actions of activators and enhancers in DNA transcription. And so the first thing that happens is that these activators bind to the distal control elements. 3 of these distal control elements make one enhancer. So then the DNA bending protein attaches to the DNA sequence, And so now these bound activators are brought closer to the promoter. And so the next step is that the mediator proteins are attached to these bound activators, and then the general transcription factors are also attached. And finally, RNA polymerase is attached to allow for mRNA synthesis. So this whole thing is called a transcription initiation complex, and this allows for the synthesis of mRNA, and therefore more abundant protein is able to be produced.